A very good afternoon to you all and welcome to our biology class of today. My name is Teacher Pacific. Today we are going to discuss on photosynthesis and leaf structure, but before we start, I hope you had enough time to watch fully our previous videos on biological molecules and enzymes. Now today we're discussing on photosynthesis and leaf structure. I've put together everything I believe that we need to know on this very topic. That includes our video. We see some questions that were asked in the previous years in IGCSE international exams, and as well some videos that are going to help us understand the topic better. So make sure you're seated in a place with your book so you get to understand everything. And in case there is a challenge, you can feel free to reach out to me on this email and I reply right away. So in our today's topic, we talk about photosynthesis. We get to understand nicely photosynthesis. We talk about chlorophyll, the use and storage of carbohydrates. We discuss minerals in plants. We see how we can investigate the needs of chlorophyll, light, and carbon dioxide, and also investigate the rate of photosynthesis, investigating the gas exchange, photosynthesis, chemical equations. We see the limiting factors of photosynthesis. We see the leaf structure, that include the external and internal structures of it. And as well, we identify the leaf structure in a dicotyledonous plant. So I hope you're ready. Make sure you have your book, you sit and watch our video fully so we get to understand everything that we need to know. Now, photosynthesis, before we even start another thing, we understand we have in two words. We have photo and also we have synthesis. Usually photo means light. It is not like a picture we usually talk about. Photo means light. Now, synthesis means making. It's like we may say light making. Now, green plants make carbohydrates. In biological molecules, we talked about these carbohydrates. We saw types being glucose, which is a monosaccharide. We talked about the saccharide, like maltose. And also we saw polysaccharides, like cellulose and others like starch that we talked about. Now, for photosynthesis to take place, we need what we call raw materials. And in raw materials, we see two raw materials, and it is the first thing that you can even write in the book. Because green plants make carbohydrates, that means glucose. This is the food. In primary, when we are defining photosynthesis, we usually say that this is the process by which plants make their own food. But now here in IGCSE, we say a different thing. We can talk about a plant making its own food, but which type of food are we talking about? Means this is the food we're talking about usually. Means they make glucose. From raw materials, there we have carbon dioxide and water. Means carbon dioxide and water, these are the two raw materials needed for a plant to make its own food. And the food that we're talking about here, it is carbohydrates. At the same time, oxygen is made. It is another product. Means we saw two raw materials. The first one is here. And also we have another raw material, which is water. The products, we have oxygen and we have glucose. Now, the reaction requires energy, which is obtained by the pigment chlorophyll, trapping the light from the sun. And chlorophyll, we know this is a green pigment, which is inside the chloroplast. And the function of this chlorophyll is to absorb light. So, photosynthesis is defined as the process, not like the process by which plant makes its own food. It is, of course, that, but here we add on something. That means photosynthesis is a process by which plant manufactures carbohydrates from raw materials using energy from the light. This definition in the post-paper question, we see that it is given 3 marks. A process by which the plant manufactures carbohydrate, 1 mark, from raw materials that we talked about using energy from light. The 3rd mark is right here. Means if we talk about the carbohydrate which is manufactured, this is glucose. The raw materials which is needed, we saw that it is carbon dioxide and water, and also we saw that it needs the energy from light which is absorbed by the chlorophyll, like we're from saying. I hope it is clear here. All right. Now, here we have our plant. Now we can ask ourselves where does the raw materials come from and where do the products go? If we saw the raw materials, we have carbon dioxide. This diffuses from air and enters in the leaf. All right. And also, with the second thing we need is water, and water is taken in from the soil by the roots. Now you understand, if water is there in the leaf, and of course water will move all the way in the plant via xylem and enters the leaf. Now our leaf is ready to do photosynthesis because all the needed raw materials are there. We have carbon dioxide that diffused in the stomata, enters the plant, and also we have water which is taken in from the soil and goes all the way in the stem via the xylem vessels until it reaches in the plant. Now you can see that our plant is ready to do photosynthesis. Now, photosynthesis, as it happens, we saw that we have two products that are going to be released out. We have oxygen, which is going to be released out, to be given out, and also we have glucose, which is going to be transported in different parts of the plant. Here we need to understand that the moment glucose is made, it's turned into starch. Why? Because glucose is very reactive and soluble, but this starch is insoluble. So always the plant decides to store its food in form of starch. We see that. So, but if starch can be stored, like examples that we saw, starch can be found in potatoes, which is always here. So you understand, if glucose is made here in the leaf, I want us to understand it very well. Glucose is turned into sucrose. That's the way glucose is transported. It's turned into sucrose, and sucrose will be transported via the phloem from the leaf. The leaf now is the source, and this one is the sink because it is where the food is going to be sent. So understand, what is made is glucose, and it is going to be turned into sucrose, and it's transported via the phloem all the way to be stored to the sink right here. Now we understand that our glucose, which was transported in sucrose form, will be stored in starch form, which is a polysaccharide. So we keep on repeating those three words, get to memorize them. So we can talk about glucose, sucrose, which is the way glucose is transported from the source where it is made to the sink where it is sent, and also understand that glucose is stored as starch. Why? Because starch is insoluble and less reactive compared to glucose. I hope that one was also clear. Now, examinative and tricks. If asked the raw materials, I told you it is carbon dioxide and water, never light. Light is not a raw material, all right? Although it requires for the reaction to take place, light energy is not a substance, therefore cannot be a raw material. So we have two raw materials simply. We have carbon dioxide and we have water. So there are two right here. Now, photosynthesis, water equation, we should memorize it. And we see that it is the opposite of respiration, if you may say. So we see that we need carbon dioxide and water. These are raw materials. And we have glucose and oxygen, which are the products of this chemical reaction. But of course, we also need light energy, which is absorbed by chlorophyll. And chlorophyll, which is found inside the chloroplast. I think it is clear. So they ask you this to write the word equation, but there are times also they can ask you to write the chemical equation, so you should be ready for the two. But understanding that the raw materials are two, let no one ask you the raw materials, and we say that it's light, yet we see a light is not a raw material. All right. And here we have it. Carbon dioxide, where does carbon dioxide come from? It's exactly what we're talking about. Diffuses into the leaf via the stomata. All right. Now here, inside here we're in the leaf, because it's where photosynthesis might happen. I'm just giving an example. So if it happens in the leaf, now we see that diffuses via the stomata. Carbon dioxide is there. And also we have water, which is taken up by the roots and transported through the xylem to the leaf. Now we see that our leaf is ready for photosynthesis since the two raw materials are there. Chlorophyll, it's a green pigment. It also absorbs light. So we should understand that photosynthesis happens when there is light. The moment there is darkness, now respiration happens. But photosynthesis really needs light. So the products, we see that we have glucose. Glucose is one product which is used to make substance needed by the plant. And also it's used in respiration to release energy. Remember, the plant also does respiration mostly nighttime, so you should understand that respiration also needs this glucose which is produced. And of course, glucose can be used for other purposes in the plant, a lot of them that we will see in our next slides. And also we have oxygen, which is a product which diffuses out of the leaf as a waste product and is also used in respiration, like you may say. So understand where the raw materials come from and where the product goes. We should understand that what this glucose is used in the plant, in respiration, to give energy to the plant and all that, but also oxygen, some diffuse out, another one is used in respiration in the plant, because also the plant does respiration. Now, chlorophyll, like I said, this one is a green pigment which is found in the plant and in the chloroplast. It's exactly what it is. It's a green pigment which is found in the chloroplast. It reflects green light, so it is green in color. So you should understand that chlorophyll absorbs light energy, and its role is to transfer energy from light to chemical energy, a very important part here. This is why you can pause our video right here and you copy this at the back of your book. Understand the role, the main role. It absorbs light, of course, but why does it absorb light? It can convert this light into chemical energy. For the synthesis of carbohydrates, such as glucose. So the same thing that we have. Understand the role of this one. It of course absorbs light, but why? Because it converts light into chemical energy, and this can help for the synthesis of the carbohydrates that we have here, such as glucose. All right. I also want you to pause the video right here and write this. How are the products of photosynthesis used? A question which is always asked out of 5 marks. The moment glucose is there, what is the role of this glucose? What is it used for in the plant? The first thing, understand that it is converted into starch. Being converted into starch, we saw that this is the storage form. It is stored. Glucose is stored in starch. This is why if I even want to check if a plant is doing photosynthesis, it is very hard for me to test glucose. I test starch because if I found starch there, it is a clear evidence that this plant has been doing photosynthesis. Because glucose was produced, then converted into starch. Like I kept on repeating the reason why plants convert glucose into starch. The reason, it is because starch is insoluble. It is less reactive. And the moment the plant needs energy, we convert the starch again into glucose. But glucose which is produced is converted into starch. This is the first use of glucose in the plant. Molecule which acts as effective energy store. Glucose which is produced can be formed into cellulose. Like you remember, we keep on saying that cellulose, it is a polysaccharide. It builds a cell wall. We know the cell wall of plant cell is made up of cellulose. It's a polysaccharide of glucose. Means if glucose is produced, it can be converted into cellulose to build cell wall. The third part, it's from what I'm talking about. Also, glucose which is formed can be used in respiration to provide energy, mostly in nighttime. Nighttime, because you understand the plant has glucose, it has oxygen, so it can do respiration. Because the moment there is light, photosynthesis happens mostly. But in the dark, a plant can do respiration to get energy and everything. So the glucose which is produced can be used in respiration and provide energy for the plant. A very important point, this one right here, maybe which is new to many people. Glucose which is formed is converted always into sucrose for transport in the phloem. And I told you it comes from the leaf, okay? It can come from the leaf and goes all the way to the roots. Means the leaf at that time is the source. There is something that we call the source. The source. And this glucose will be sent like to the roots, which is the sink. We learn it maybe in our next video. But understand that glucose is always transported in form of sucrose and stored in form of starch. And you should understand that starch is less reactive. Sucrose is less reactive compared to glucose as well. So that's why the moment glucose is made, it's turned into sucrose and it is transported and stored into starch. This is the fourth point of what glucose is used for when it is produced. The fifth point is that glucose can be used as a nectar because it is sweet to attract insects for pollination. Means it can attract different pollinators. So here are the 5 points that you talk about. These are the very things that glucose is used the moment it is produced. Means it can be converted into starch, which is the effective energy store. It can be converted into cellulose to build, because cellulose is the polymer of glucose to build cell wall. Glucose can be used in respiration to provide energy, and also it can be converted to sucrose for transport in the phloem, and at the end it can be used as a nectar to attract insects for pollination. So these ones, I hope you guys will pause here and write them down. Now, most important thing as well, plants can also convert carbohydrates they make into lipids. Means plant can convert the other glucose, which is a carbohydrate, into lipids for energy source in the seed, and also into amino acid. By the way, we saw that glucose which is produced is transported via the phloem in form of sucrose. Means sucrose can be transported in the phloem. Also amino acids can be transported into phloem. We should know this one also can be transported. So understand that the carbohydrates can be made into lipids for energy store in seeds, but also into amino acids, which is used to make proteins when combined with nitrogen, and also other mineral ions absorbed by roots. This one, we talked about it in a previous video. So we understand that elements that makes carbohydrate, it is C, H, and O. Remember, there is no nitrogen because nitrogen is only found in proteins, in amino acids. So we understand that for us to convert carbohydrates into amino acids, then we need to combine it with nitrogen, so it can be added there as well. So if you followed our previous video, I think this is very easy to understand. All right, as I told you, we have some questions we are going to be seeing in our presentations that were asked in the past paper. Starch and sucrose. This is the storage form, which is effective. And also we have the sucrose, which is a transport form. This is exactly how glucose is transported. It means the plant turns glucose into sucrose, and sucrose can be transported from the source where this glucose is made to the sink, to where it is sent. Are made by plants after photosynthesis. State the name of tissue that transports sucrose around the plant and name the other biological molecule that is transported in this structure. Which tissue by which transports sucrose? This one you already know it. It is the phloem. And we have two tissues, by the way, in transport. We have phloem that can transport the sucrose and others, but also we have xylem that can transport water. So as we talk about them, make sure you even understand the adaptation. Like xylem is very hollow, and it is open end to end and strong to give support to the plants. We should understand that. But the tissue here for sucrose, since it's the one we are talking about, it is phloem. Another biological molecule is amino acid, because I told you for phloem, it is that one that we are talking about, phloem. And also, we have a biological molecule that we can talk about. That one will be amino acid, because both amino acid and sucrose can be transported via the phloem. Describe how starch is broken down in the human digestive system. This one, I hope we will get time to talk about digestion, which is a video I'm sending tomorrow at around 11. So we get to see also digestion because it's one of the sensitive topics. But for this one, the human digestive system, if we talk about the way starch is digested, we understand that it's digested, we start from the mouth and also in the small intestine. Like I told you, we need enzymes to break down these bigger molecules. That is amylase, which converts starch into maltose. Then we need maltase, which is going to convert maltose into glucose. So I hope the two will help us understand better what we'll be talking about right here. I hope they are clear. Now, going to the next point that we'll be talking about, we saw the moment glucose of ours is formed, we should understand what it's turned into. So, glucose can turn into yes, as you can see for glucose, which is formed like from saying, it can be converted into different substances. Like the first one, you can see that the one that we have here can be converted into fats and oil. That's the lipids that we talked about. It can be converted into sucrose for transport around the plant. It can combine with nitrate. Nitrogen, remember, it is what we are mostly interested in, and convert into amino acid. It can be converted into cellulose, which is a polysaccharide, and it can also be converted into starch, which is this for storage, which is insoluble, like we said, but won't affect osmosis in cells. Now, minerals in plants, when I talk of minerals in plants, we know different minerals. But the most important minerals that we talk about, it is magnesium, that I want us to talk about, and this nitrate right here. Now, for this plant that we have right here, it has full nutrients. The nutrients that we talk about, we know different nutrients, but the most important part, I want to talk about minerals mostly. I want to talk about magnesium. This magnesium needed to make chlorophyll. Means a plant without magnesium won't make chlorophyll. Means chlorophyll needs to be absorbed, to absorb light for photosynthesis. Without chlorophyll, you understand this plant won't be doing photosynthesis. Magnesium is very important to make chlorophyll. You can also pause this video and write it. It's very important. Nitrates, they are needed to make amino acids. Remember, from what you're talking about here, we saw that if you have glucose combined with nitrates for us to have amino acids. That means without nitrates, a plant of ours, this mineral is very important, because without nitrate, then a plant don't have amino acid. Amino acids are needed to make proteins for growth. That means a plant without nitrates won't grow very well, because there won't be proteins to help it to grow. Now, we see that if we talk about deficiency of these minerals, like magnesium ions not being there, that means there will be yellowing between veins, and it is what you call chrolysis. Chrolysis, it is when this plant seems to be yellow, that means there is no chlorophyll. If you don't have no chlorophyll, then no photosynthesis. I think that one is clear. By the moment also this plant don't have nitrate, of course it will be yellowish, because it won't be making photosynthesis like we say, but as well it won't be growing very well. So they are going to be short plants, and you understand the production of them can be limited. So talking about the minerals, get to understand this magnesium needed for chlorophyll. Without chlorophyll there will be chrolysis. The leaves will tend to be yellow. That means the food won't be made like in a way they should be made. It won't be making photosynthesis, so it will be having a silent growth. If you talk about nitrate, understand the formation of amino acid. Without amino acid, no proteins, so no growth. Means magnesium needed for chlorophyll, for photosynthesis to happen, and the plant makes it into one food. Nitrate not being there, we won't see growth. There will be a stunted growth. Magnesium not being there, yellowing of leaves. So I hope it is clear and we understand the difference between the two. And it is one of the most asked parts on these minerals, mostly these two, magnesium and nitrate. Growth, chlorophyll. So it is here. So magnesium is needed to make chlorophyll. Nitrate is needed as a source of nitrogen, needed to make amino acid, to build proteins which help in growth of the plant. That means deficiency of magnesium put yellowing between the veins, so we understand there will be chrolysis, but also nitrate not being there, it will cause a stunted growth. This plant won't grow very, very well. I repeat again, very important to understand why nitrate is needed. Nitrate is the source of nitrogen, and nitrogen makes amino acid. As we know, glucose has C, H, and O. These are the only elements we saw in biological molecules that are in carbohydrates. I'm repeating this because I talked about it. Nitrate, we hope it to have nitrogen also. It's the source of nitrogen. Now, if you have CHON, now we can have amino acid. Because this amino acid is the only one with nitrogen. Lipids don't have it. Remember, even carbohydrates don't have nitrogen. But if nitrate comes to carbohydrates, then we are going to have amino acids. You should be able to explain this very well. Understand the reason why nitrate is needed is to make amino acid to build protein which help the plant to grow. Deficiency in nitrate, it won't grow very well. We have a stunted growth. Now, talking about deficiency of magnesium, we understand because chlorosis, which leads to plants being yellowish. So it's exactly what you're talking about. This is a healthy plant. It has everything. But the moment this plant lacks magnesium, that means chlorophyll, which is a green pigment, won't be there. We are going to see chlorosis here. We can see the yellow part. This one means we don't have chlorophyll. Why? No magnesium. And they ask it like this. We even see many other questions. Tell the name of one mineral that is needed to make chlorophyll. That's magnesium ions. Now, investigating the need for chlorophyll. Does the plant need chlorophyll? That's the question we ask ourselves here. Does the plant need chlorophyll? How do we check that? So we can check on a plant with chlorophyll, maybe with a plant without a chlorophyll. Then we see which one that can be healthy. So from what you can understand before we start investigating, to understand the plant with chlorophyll, it will absorb light and the plant will make its own food. How do we know that a plant has made its own food? We are going to see that it has starch. So we can always test starch. Means if chlorophyll is there, we are going to find starch. I will give you an example. Like on this plant right here. If you can test this plant, the whole of it, if you can test starch, I want you to follow this very nicely. If we can test starch on this plant right here, so we understand we are going to see what we do, but at the end to test starch, we use iodine. If you use iodine, I want to show that this place here, which is green, which means it has chlorophyll, can turn into blue-black. But this place here, which is yellowish, cannot turn into blue-black. Why? Because we don't have starch here. If there is no chlorophyll, we can't find their starch because photosynthesis will not happen. I want to think that you understand it. Okay. How do we test that? Means starch is stored in chloroplasts where photosynthesis occurs. Means testing leaf starch is a reliable indicator of which parts of the leaf are photosynthesizing. Means if I find the place can turn into blue-black, then I can know photosynthesis happened there. How do we do it? I want you to follow it nicely here. Now, the first thing that we do, we put a leaf in boiling water. And why do we put a leaf in boiling water? Why do we put a leaf in boiling water? It is to break down the cell membrane and the cell wall. It is the first thing that we do. You cannot test for starch and you bring a leaf and you put iodine on a leaf. That iodine can't easily go into. But what we do, we first break down cell wall and cell membrane. It means we put a leaf in a boiling water. That's the first thing. After that, the leaf is put in hot ethanol. Why do we put in hot ethanol? This removes chlorophyll. Usually, chlorophyll cannot dissolve in water. That is impossible. But we put a leaf in ethanol. Ethanol, which is boiling, dissolves chlorophyll. So we understand the first thing, we broke down the cell wall and the cell membrane. That's the first thing in boiling water. The second thing we do, we remove chlorophyll, all right, by putting it in ethanol. Then we dip it in boiling water to soften it again. After removing all the other chlorophyll, then we soften it. These three procedures are important because they always ask this question. They tell you, why did they put a leaf in boiling water? You should understand it is to break down cell wall and cell membrane. Why did they put it in ethanol? To remove chlorophyll. And why did they put it back in boiling water? It is to soften it because now it is a bit hard by that time. Now, after that, we understand the three processes we use. The first one, we put our leaf in boiling water and to break down the cell wall and the cell membrane. Then we put it in ethanol to dissolve and remove the chlorophyll. Then we put it back in boiling water to soften the leaf. Now, by that time, starch is exposed. Then we can put it out in solution. In a green leaf, the entire leaf will turn blue-black. As photosynthesized, it means if the whole leaf is blue, we are going to see that if you put it in iodine after the other three steps we talked about, we see that it is going to all of it turn into blue-black. But now, there is a leaf here which is called variegated leaf. Variegated leaf is like this one right here. What is the meaning of this one being variegated leaf? We can see that we have the whole leaf that we're seeing. We have where we have yellow. Where we have yellow means we don't have chlorophyll. And it is what we are going to use to check if chlorophyll is really important on the leaf. The whole leaf that we have here, if, as you can see it, maybe if you can see this one here, this green part, after we've boiled it to break down the cell wall and cell membrane, after we've put it in alcohol ethanol to remove the chlorophyll, after putting it back in water to soften the leaf, now we understand chlorophyll, our starch is exposed out here. We can put on iodine. If you put in iodine, we expect this part right here that you see here will turn into blue-black. But this one out here won't turn blue-black when we see it. Why? We don't have chlorophyll, so photosynthesis don't take place in this yellow part right here. Now, we are done with the first part. Now, this part can also be tested whether chlorophyll is needed by using a variegated leaf. One that is partially green and partially white, like we're from seeing, means the white area of the leaf contains no chlorophyll. So when the leaf is tested, only the area of chlorophyll turns blue-black because it is where we have starch. The area that had no chlorophyll remained orange-brown, which is the color of iodine. So no photosynthesis occurred here. And there is no starch stored. So I hope you're understanding the difference between the two parts. Get to understand it and be able to explain it very, very well. So with that being said, I want us to follow this video right here, which is going to help us to understand it better. All right. So this is the one that I want us to check, this video right here, on the need of chlorophyll for photosynthesis.
Destarch a variegated plant by putting it in the dark for at least 24 hours.
So when we talk of destarching, is like removing starch. If you put a plant somewhere there is darkness, so you should understand that it is going to use the starch it has been with for respiration, right? Because it can't do photosynthesis to make more glucose to form more starch. So destarching means if you put it in a dark place, it will use starch to turn it into glucose, then does more respiration, right? So destarching is like using the starch that it had been with.
Pick a leaf from the plant and boil the leaf in a beaker of water for a minute.
Like we said, remember, putting it in boiling water, I think that should give us the meaning of it. So I hope you remember what I'm from saying. It is to break down the cell wall and the cell membrane, and that is a variegated leaf, since we see that it has some parts which are green and others which are not green. I hope you understand what is the purpose of boiling the leaf in water. You understand, to break down cell wall and cell membrane. That's the first thing we do, because starch, remember, is inside. It is not outside the cell part.
Turn off the Bunsen burner. Transfer the leaf to a boiling tube half filled with absolute alcohol and put the tube into a beaker of hot water.
What is the purpose of boiling the leaf in absolute alcohol? For us, we use the ethanol in our presentation. So the reason is to dissolve chlorophyll. I told you this chlorophyll can only dissolve in alcohol, like ethanol. So that means we are removing the chlorophyll. What tells you that chlorophyll can dissolve? You can even see the color of our alcohol even turning green. So I think it's a bit clear there.
When the leaf has decolorized, transfer it from the tube to the hot water again.
All right. Putting it in hot water again, like we are from saying, it is to soften the leaf, right? Because our leaf is already now strong, because we are from removing chlorophyll. So we're putting it back here for it to be soft. Now our leaf is ready to test if there is a starch in it.
Spread the leaf on a white tile and add a few drops of iodine solution onto its surface. Observe any color change in the leaf.
Why do we need to test a leaf from the starch plant for starch? So that one we should also ask ourselves on that, right? So because if you remove the starch, then why? Why? That one
Expose the whole plant to bright light for about 5 hours. Pick a leaf and draw a sketch of it to show the color pattern.
Remember, this is our leaf. We can see we will expect the color to change here. All right, I hope you agree, because we saw the need of chlorophyll. If chlorophyll is there, that means this part here can make photosynthesis, and we can find starch there. But here, I don't think we can find any starch here. I think it is clear. So, as from what you did on testing starch, even on the color we're expecting to have blue-black right here, but you don't find it here.
Test the leaf for starch.
All right, I think you're seeing exactly what we're talking about. So this shows the need for chlorophyll in photosynthesis. Where we have chlorophyll, starch can be there. If we find starch there, that means photosynthesis only happens where we have chlorophyll. So you can see, and it was green, but here it's a clear indicator that we don't have chlorophyll, simply no photosynthesis. Why? Because we don't have starch. Starch is always there when photosynthesis happens.
Draw another sketch of it to show the result of the iodine test.
Yeah, here is the results. Where this before, we had green and this non-green. That means we have chlorophyll. Where we have chlorophyll, we can see that starch is there because our brown color turned into blue-black. But here means we don't have starch. If we don't have starch, means no photosynthesis happened. I think it is clear. This is the test that we do to show the need for chlorophyll in photosynthesis. I think it is very clear. And we use a variegate leaf so it can show us the parts. Where we have green means we have chlorophyll. And we saw that where we have chlorophyll is where photosynthesis happens. Why? Because we have starch there at the end. I think it is clear. So on our drawing, I think you can see it here. We boiled the leaf. I hope you saw we boiled to break down the cell wall and cell membrane. Then we put it in ethanol. Why? To remove chlorophyll. Then ethanol turns green. That shows that this chlorophyll can dissolve in ethanol. And now we can see we wash it. Washing it, we saw it is to soften it, to make sure the leaf is soft. Then if you put on iodine, it will turn into blue-black, where we had the green before. Now, investigating the need of light. Do we really need light for photosynthesis to happen? Here also we can find out by doing a simple experiment of putting a plant where there is light and where we don't have light. And we see where we can test starch and we find. If our leaf is where we have light, so we understand we test chlorophyll and we find it, starch and we find it there. If we put it in the dark, we understand no photosynthesis. We won't find their starch. Now, before starting the experiment, the plant needs to be destarched. We saw that we can put it somewhere dark. And if you put it somewhere dark, you understand respiration will happen and use the starch of ours. This is to ensure that any starch already present in the leaf will be used up. This is why we first destarch. And now we go to test again. All right? Because if we don't destarch before, then we are going to use the starch that has been there. But we need to have a leaf without starch. One, we put it where we have light. Another one, we put it in the dark. Then if we do that, we see where we have light if we can form more starch, or we see where the one in the dark if we can make starch. I think it's clear. It's clear. Means any starch already present in the leaf will be used up and will not affect the result of ours. This one is one of the most asked points. So it means you pay attention to it. You are going to test if starch is present, but you are distarching. You want to make sure all the starch that has been there is used up. Now I put it on light, another one I put it in the dark. Now after some time I check if we have starch where we have light or we have it in the dark. Now following distarching, the leaf of the plant will partially be covered by aluminium foil. This aluminium foil, you understand it is to avoid any light going to our leaf and place it in the sunlight for a day. Now the leaf is then removed and tested for starch. If you test for starch, that's what will tell us that if really starch is formed or not. Now the areas of leaf that was covered with aluminium foil will remain orange. You understand if I have, let's say maybe this is our leaf. If I cover it, all right, with aluminium foil, maybe I cover this part. What we are going to expect is that if I happen to put iodine here, if I remove this aluminium here, there won't be starch. If I put iodine, it won't change any color. We'll see. But this part here, which is exposed on light, we are expecting it to turn into blue-black because with light, photosynthesis can happen. With the darkness, only respiration can happen. So, and if light is there, then it can make starch as well. Now, the area of the leaf that was covered with aluminium foil remains orange-brown as did not receive any sunlight, and it could not photosynthesize. While the area exposed on the sunlight, we turn blue-black. It's what we are talking about here. This proves that light is necessary for photosynthesis and production of starch. I want us to check on this video here so we get also to understand it better. All right.
2 to 3 days. This step is essential in order to rid the plant of any starch that was formed in the leaves prior to the start of the experiment.
Like we said, we start by distarching. If we put it in the dark for 2-3 days, distarching happens because in the dark, a plant does respiration. If a plant does respiration, it means it is using up all the starch which is there because starch is turned into glucose, then respiration takes place for the plant to get energy. All right? Then we distarch always to make sure all the starch that has been there is used up.
Experiment. At the end of this time period, remove the potted plant from the dark. Cut a piece of black paper, like so, and secure the cut end of the paper using a sticky tape. To make this experiment a bit more interesting and fun, I have carved out my initial, which is the letter T as well, on the black paper covering the leaf.
So we are going to take maybe one leaf, we put it inside here. So we see one part of the leaf will be in the dark, another part will be exposed on the light. Now at the end, we are going to test starch. We check if whether where we had light photosynthesis happened, that we know it by finding that starch.
So, and secure the paper on the leaf using some paper clips.
It's exactly like this.
Potted plant in sunlight for several hours. Then, detach the leaf from the plant and remove the black paper from the leaf. Now, introduce the leaf into the test tube containing the alcohol solution.
Remember, we are putting it in alcohol solution to make sure that we remove the chlorophyll.
Place the tube in the boiling water bath. If you're using an open flame such as spirit lamp or Bunsen burner for your water bath, then make sure to turn off the burner flame once the tube has been during the boiling process, which is a huge possibility considering the highly flammable nature of alcohols. Alternatively, you may use a hot plate for boiling like the one I'm using here instead of an open flame. During this boiling step, chlorophyll pigments in the leaf are decolorized by the hot alcohol solution, resulting in a bleached leaf with pale white color. This step is necessary for a better visualization of the iodine-starch reaction on the leaf surface.
Like you can see that our alcohol is changing the color to green. That means it is dissolving what? It is dissolving the chlorophyll. And remember,
For the
we're also
leaf in a
putting it
petri
in,
dish
we'll put it again in water to make sure we
containing
soften it.
iodine solution. After a couple
Now we are putting our leaf in iodine solution. We're expecting the color to change. Remember, we had a tea-like structure. That's where light was exposed.
of minutes, you will see that the exposed parts of the leaf not covered by the black paper turn blue-black in color in the iodine solution.
I think you can see it here. You can see the other T's here. Now, if the color turns into blue-black, you understand we have starch in here. It means here the plant made its own food. That means there is a need of light for photosynthesis to happen. Where we didn't have light, we can see the color that we have right there. I think it is clear, right?
The part which was covered by the black paper, however, do not show any significant change in color except for a faint brown iodine color. This is a negative test for starch, and
I think it is clear now that we can see it very well. Where light was exposed, we can see that there is that. It means we need light for photosynthesis to happen. You should be able to explain these processes by explaining why the color is changing. Remember, that's the taste of starch. We should be able to explain why we are boiling the leaf, why we put it back in water.
thereby photosynthesis. This simple experiment clearly demonstrates that light is an essential external component for
Light is very essential for photosynthesis. It means without light, you can see there is no photosynthesis.
photosynthesis to take place in a plant.
Yeah, that's it. So these are the three conditions that you're talking about that's always very important for photosynthesis. I started on what? We started by talking about chlorophyll. Uh-huh. We talked about light, which is also a need. And at the end also, we can see that we have...
This is a 3. It's sloppily written and rendered at an extremely low resolution.
Yes. We saw the need of light. We tested it. We saw how we test it. We can put it in light and we cover it with aluminum foil. Then we test. We see the place that we were covered and the exposed one, where the light is needed for photosynthesis and all that, and also investigating the need of carbon dioxide now. Is carbon dioxide needed for photosynthesis to happen? That one also can be tested by putting one plant where there is carbon dioxide, where we don't have carbon dioxide. So you understand that will be our independent variable. That's exactly what we changed, investigating the need of carbon dioxide. Now we see that the one where we have carbon dioxide, there is starch. And we can expect where we didn't have carbon dioxide, that there was no starch. That can be our prediction. We are going to start by, like always, destarching. We saw the reason why we destarch. We destarch to make sure that the starch that has been there affects our current investigation. 2 plants by placing them in the dark for a prolonged period of time. Place one plant containing beaker of sodium hydroxide, which will absorb carbon dioxide. That is something we need to know, the need of carbon dioxide. It can absorb carbon dioxide from the surrounding air. Another one, we put it where we don't have something that can absorb carbon dioxide. Means one plant, we give it carbon dioxide, another one we don't. Anytime we talk about sodium hydroxide, it is always there to absorb carbon dioxide. So that means we don't have carbon dioxide there. Then we place them in the bright light. This will be our control variable. Both plants should be where we have light. Enough light, by the way. So we aren't changing one variable. Otherwise, it can't be a fair test. Now, we test starch for both plants. But we see the leaf from the plant placed near sodium hydroxide will remain orange. You understand? We don't have carbon dioxide, so that one has to remain orange. That means we don't have starch. Could not photosynthesize due to the lack of carbon dioxide. The leaf from the plant placed near water should turn blue-black, as it has all necessary requirements for photosynthesis. And here we have it. If you see this sodium hydroxide, it is to make sure it absorbs any carbon dioxide. That means this plant don't have carbon dioxide. But this one does have carbon dioxide because it's just placed in water. Means if we test their leaves after some time, for starch, we find that this leaf right here has starch in it. But this one won't be having starch. Like always, you are going to do the same thing. We destarch, but after, we are going to boil our leaves, both of them after some time. We put it in them in alcohol to remove chlorophyll. We put them back in water to make them soft. Then we put iodine to both of them. We are going to see that there won't be any color change here. It will keep on brown because carbon dioxide is very essential for photosynthesis to take place. Now, another investigation which is very important, and it is ever in paper 6 that we need to check on as well. What is the rate of photosynthesis? We understand that. How do we measure that? Here, what I focus on is the dependent variable. What do I measure each and every time they ask me to measure the rate of photosynthesis? I have to measure the bubbles produced over time. Even when they ask you to make an investigation, like an equation, right? They ask us to do that. Make sure you know you are a dependent variable. The dependent, it is to measure the volume or the amount bubble produced over 1 minute. Because the rate, if someone talks about rate and you don't talk about time, then you understand it's going to be wrong, obviously. Now, the more bubbles produced per minute, the faster the rate of photosynthesis. But now, a more accurate version of this experiment to correct oxygen released in a test tube inverted over the top of conduit over a long period of time, and then we measure the volume of oxygen corrected. Understand this is exactly what you measure. If someone say investigate the rate of photosynthesis, what do we measure? We measure the bubbles. That's the only outcome that we measure here. Over time, since we are talking about rate. And we can see it right here. For example, if you're investigating the effects of light intensity, this one is enough. Reading this sentence right here should tell me the independent variable. I want everyone to follow because this is a very important question. If they say investigating the effects of changing light intensity, you understand this is the independent variable. We are changing light intensity. What are we going to measure? Nobody needs to say that. We are measuring the volume of gas produced per unit time, if it is rate of photosynthesis. If you say investigate, what do we keep the same? Because light is the only thing we are changing, means we should keep the same. The temperature should keep the same. Carbon dioxide should keep the same. Those ones are going to be our control variable. But if someone says investigate the effects of changing light intensity, you're only changing light intensity. Temperature should be the same. Carbon dioxide should be the same. Amount of water should be the same. I'm only changing the light intensity. What is the dependent variable? What we are measuring? We're only measuring the volume of carbon dioxide produced per unit time because we are talking about rate. We can say, if someone says what the dependent, it is volume of oxygen produced in 1 minute. If you don't put on time, that won't be rate you're talking about. This experiment right here, there is a video I want you to follow very well so we get to understand how it is done as well. All right, here it is.
Count the number of bubbles of oxygen gas we see leaving that cut stem. So I just need to get down here and use my dissecting scissors just to...
So the experiment we have here, we are going to investigate the effects of light intensity. We are going to change the distance. This is why you are seeing this ruler right here. We are changing the distance of this lamp, which is here. So, and we check, we measure. We measure the amount of bubbles produced. That is if they say investigating the effects of light intensity on the rate of photosynthesis. We are going to use per unit time. Means every minute we'll be measuring the amount of bubbles that we collected.
Snip. There we go. The very end of my pondweed off. Just going to push that a little bit lower in my solution. There we go. So now I'm going to be able to count the number of bubbles of oxygen produced in 1 minute, and that
we are going to be measuring the amount of bubbles produced in 1 minute. You understand? 1 minute, it's rate. Anytime you're talking about photosynthesis, the dependent variable is always going to be amount of gas produced. That can be bubbles over time. You can use a syringe and you measure the volume of gas produced in 1 minute. But whatever you do it by unit time. All right? If you forget that by 1 minute, then you're not talking about rate.
Will give us a measure of the rate of photosynthesis. So I've cut my pondweed at an angle across the stem because that's the best way of ensuring you get a nice steady stream of oxygen bubbles coming from it. So the independent variable is going to be our light intensity. And
independent variable is light intensity. So you are going to be changing the distance by using that ruler.
Change that. We're going to change the distance distance to be our light intensity, and to change that we're going to change the distance between our pondweed and our light source. Now I'm using quite a powerful light bulb here, but actually it may be better, and you may have done this at school, to use an LED light source. This is because the LED sources don't give off very much heat energy, and of course because the temperature will also affect the rate of the photosynthesis, we don't want to have two variables changing at once because that would make the test unfair. But this is the one I'm using today.
So I hope you understand the role of using LED lights, because this one can give out light because we are changing light intensity, remember, okay? But the problem with these ones also, they might give out heat. And if it gives out heat, now you understand we have two variables we are changing. And to make a fair test, only one variable should be changing, all right? Means LED light can only change light intensity. But these ones can change light intensity and give heat out. And we don't want heat. We want heat to be the same. We want carbon dioxide to be the same. We want everything else to be the same. And we change only one variable, which is light intensity. So for us to have a fair test, only one variable should be changing.
And so this is the one I'm going to investigate. So the end of my ruler, I've moved in so it's directly in line with the button. So it's aligned to the 10 centimetre mark on the ruler. So now there's a 10 centimetre distance between the light source and the pondweed. I can now turn on my light. Now I'd want to turn the overhead lights off at this point as well, so the only source of light on my pondweed is coming from my lamp directly. I'm not going to take my measurement straight away because the pondweed needs a little bit of time to adjust to the brightness of the light and start the photosynthesis at the correct rate. So I'm going to leave it just a few minutes and then I'm going to take my measurements. So I've waited a few minutes now and the pondweed has adjusted to the level of light that it's getting from the light bulb. And so now using my timer, I am going to time for 1 minute and count the number of bubbles. And that's just the number of bubbles coming from the hot surface. There may be other bubbles coming from the leaves around it, but just to keep it a fair test, I'm going to count the ones from the end of the stem. So,
remember why we are using that stopwatch? We are making sure we measure the number of bubbles produced in 1 minute. And that's what rate is about, which is our dependent variable. The independent variable, we are changing the distance between light and our pondweed.
Moving down to eye level, so I get a really...
So I think you can see the bubbles that are coming out there. They are there.
So with the pondweed, this distance from the light, we've got...
And you understand those are bubbles produced because of photosynthesis, of course. I think if you are seeing it.
To change the value for our independent variable, so I'm going to move my pondweed away from my light so that it is now 20 centimetres.
I hope you're also seeing we are changing the distance, all right? We change the distance, which is our independent variable. We're changing the distance between the lamp and our pondweed.
From the light. New level of light, and then I will count the bubbles again. Now I need to do 5 values for my independent variable, so I'll repeat the experiment with the pondweed at 30.
We are keeping on adjusting, so this is our independent variable. Now we change the distance between our lamp and our pondweed because it is the independent variable. Then as we change, we wait for 1 minute, we record the number of bubbles in 1 minute. Means what you are changing is the distance of it, which is the light intensity. What you are measuring, it is the number of bubbles in 1 minute, like I'm saying. But other things should be the same there. We should make sure we have the same carbon dioxide around, we have the same amount of water, we have the same species of pondweed and all that.
Results will look a little bit like this. And as you can see...
I think you can see we are increasing the distance. Means the further we go from our light source, we can see that the number of bubbles in 1 minute, they are reducing. That means the closer we were, we produced more bubbles, which says the rate of photosynthesis was high, of course.
As the distance between the lights decreased. There is another way that you may have seen this experiment being done, and this is where we actually collect.
So the first way we can, the dependent variable, the thing, we can change the same thing, but we have here the same dependent variable, what you are measuring. We measured the number of bubbles in 1 minute, but here also we can use a different way to measure. All right, and it's what you have in our presentation. I want us to understand it very well. We can use this way to measure our bubbles in 1 minute, but also we can measure volume in a different way.
The gas, because of course one of the errors that we produced is exactly the same volume. So you may see it where we collect the volume of oxygen so we can measure it more accurately, or you may just collect the oxygen to prove that it is oxygen gas that the pondweed is producing. My pondweed, and I'm going to trap it under a... Now obviously again with the light source close by, the pondweed will start to photosynthesize, but to collect the oxygen I need to take a boiling tube full of water at the top of my funnel.
So we can see,
and now
we can see that the other oxygen bubbles which are being produced, we come up there, all right? We come up there, and if you want, you can close here, and we go to measure the volume of the oxygen produced. This one is more accurate, and you have to make sure you put this test tube when it is full of water, and now the more oxygen we produced, we're going to see space of oxygen up there.
As the bubbles of the tube displace the water, and then after a few hours you should have collected enough oxygen that you can either measure its volume or you can test with a glowing splint which should relight to prove that it's oxygen.
All right, so I want to think that it is clear. And it's exactly this way we have. Testing the effects of changing light intensity, we can see this is the second way she used. We can see as time goes by, as oxygen bubbles will be produced, we are going to be seeing some oxygen bubbles up here, and this is our dependent variable, and we are going to measure this volume here. We saw we should keep the temperature the same, and it is the very reason why we are seeing this glass tank filled with water. Why are we putting this water? We saw it's better, instead of using lamp, to use LED light, because only light comes from there. But if you use normal lamps, they are likely to give out light but heat at the same time. So for this one is to make sure that heat is absorbed and it is not passing, and only light will reach here. Now we have our ruler. We keep on changing the distance of which our pondweed is from the lamp. Understand the reason this one is here. It is to make sure that heat which is absorbed by this light does not pass here. Only light can come and be here. The control variable, we saw that should be the temperature. Carbon dioxide should be the control variable, means water with sodium hydrogen carbonate should be kept the same. The aquatic plant used should be the same. That's why we have control variables. What are we changing? We're only changing the distance of lamp. That's the only change, and that change changes the light intensity. I hope it is clear. Anytime they talk about investigating the effect of changing light intensity, we understand we are changing the distance of the pondweed from the light. What are we keeping the same? Temperature should be the same. Amount of water should be kept the same that we have here. The plant should be kept the same that we are testing, and all that. We should know what we are changing, what we are measuring, which is the volume, of course, of oxygen produced, and what you're keeping the same. Investigating the effects of changing temperature. For this one now, the lamp distance should be now constant. That means for us to have a fair test, we should change only one variable. Means the factor being investigated, it is we are changing temperature, all right? Because remember we are checking the effect of it, and still we are going to measure the bubbles of oxygen produced, and of course means the aquatic plant is already the same. Water with hydrogen carbonate, a control variable. Light should be kept the same to make sure that it's a fair test. We are changing only one variable. Same thing to also carbon dioxide. We are only going to change. Remember, this sodium hydrogen carbonate can be the source of carbon dioxide, all right? So we are going to be changing that, but we are measuring, as always, the dependent is bubbles of oxygen. This is exactly the only thing we measure, because that's the result of photosynthesis. Other things will be kept the same. We are changing only that one variable. We are keeping the same distance, all right? Keeping the same temperature to be a control variable. We're only changing the source of carbon dioxide. Now, alternative ways of measuring gas. We saw the best way, even in our video that we saw, We can measure the volume produced by an inverted measuring cylinder with graduated field, with water reading, which it's what we used, but also we can use syringe. And the syringe can also show us the volume of oxygen which is produced. Means as we move forward, there is also investigating the gas exchange. People, like I told you, we should expect two scenarios here and we should memorize them very well. When there is light, a plant is going to do photosynthesis. But in the dark, a plant is going to do respiration. Means if in the light, we're expecting carbon dioxide to reduce, all right? Because it is going to be used up. But in the dark, carbon dioxide anywhere will be released because there is respiration, means carbon dioxide will increase. Something we talked about, we saw that carbon dioxide is acidic. It's an acidic gas. Being acidic, you understand the pH is always going to reduce anytime we are in the dark. We are in the dark, there is respiration. Respiration, there is the use of glucose and oxygen. Carbon dioxide is released. All the time in the dark, carbon dioxide is going to increase. This means it is going to lower the pH. The place is going to be more acidic. If there is light, carbon dioxide will be used up. That means the place, whatever place we can be in or in medium, it's going to turn into basic. It means pH is going to increase. We should be able to explain that, right? Now, plants are respiring all the time. And so plants are taking in oxygen and releasing carbon dioxide by it because it's always doing respiration. Because without respiration, that's a characteristic of all living organisms. Without respiration, we understand that a plant cannot have energy. Plants also photosynthesize during daylight hours. Means in the dark, there is more respiration, photosynthesis is now there, for which they need to take in carbon dioxide and release oxygen. At night, plants do not photosynthesize, but they continue to respire because plants respire all the time. Meaning, they take in oxygen and give out carbon dioxide. That means plants sometimes release carbon dioxide. Why? Because of respiration that takes place on them. All right? And you should understand that the more we have carbon dioxide, pH. Carbon dioxide is acidic. pH will always reduce. We can see it here. Carbon dioxide enters photosynthesis, then the oxygen releases. Then for respiration, it needs oxygen for the plant to get energy. Now, photosynthesis only occurs in chloroplasts. That's something that you need to know as well. It's only occurs in chloroplasts, which are found in the following cells. We saw photosynthesis happens in 3 cells, if we can talk about. It is in the palisade cells, I think we talked about it. It is in spongy cells, and also in gourd cells around the stomata. These are the only 3 places we can find photosynthesis in plants. Remember that light is vital. Like we are from seeing in our video, we saw we can test for photosynthesis if light was used. Means if light is there, starch will be found there. If light is not there, then starch won't be there. That means even photosynthesis this happens. And also temperature is very important, but an ideal temperature, not a very high temperature, because a high temperature, we saw that it can denature. During the day, especially when the sun is bright, plants are photosynthesizing at a faster rate than they're respiring. But the plant respires all the time, right? But during the day, the light photosynthesis is more. So there is net intake of carbon dioxide and net output of oxygen because photosynthesis releases oxygen. We can investigate the effects of light on the net gas exchange in aquatic plants using pH indicator, such as this one. Also, we should memorize it. Which indicator do we use to test for photosynthesis or for net gas exchange? You should always remember this because they ask it out of 6 marks. Get to understand that carbon dioxide, when it enters, a lot of it, that photosynthesis will take place and oxygen will be released. But in the dark, we understand we have more respiration. That means more oxygen will be used and the plant will release carbon dioxide. This in this gas exchange we are talking about. We should be able to explain because it is, I think, easy to explain. This is possible because carbon dioxide is acidic. That means if more carbon dioxide is being released, and more carbon dioxide can be released when there is respiration, the place will be acidic. It's an acidic gas. So hydrogen carbonate indicator shows carbon dioxide concentration in the solution. I want us to see it in this way. Very easy to understand even if you see. Very easy to understand. That means if there is a high concentration of carbon dioxide, we're expecting more photosynthesis because it's a raw material for photosynthesis. Means the highest, we're seeing the color of hydrogen carbonate indicator is yellow. Indication in plants, more respiration, because remember, more respiration photosynthesis, and also we have lower pH. That means if you have more carbon dioxide, we understand if it is released, that means we had more respiration. Yeah. Because respiration releases a lot of carbon dioxide. If a lot of carbon dioxide is released, then we understand there is a lower pH because you saw that carbon dioxide is an acidic gas. If it is higher, then we can see the colors of them. I think it is clear. The moment you have a high carbon dioxide, if at any time someone says we have a high carbon dioxide, that means there is a lot of respiration happening. Then the pH will be lower. Why is the pH lower? Because a lot of carbon dioxide lowers the pH, it is more acidic. Atmospheric level, then we can see that they are equal. But now the moment we find that we have a very lower carbon dioxide, if it is lower, that means it was used up by photosynthesis, all right? That means we have more photosynthesis. More photosynthesis, we use up carbon dioxide. That means the place will become more alkaline. The color will be purple. In IGCSE exams, they ask this a lot. And it is very easy, by the way. It's a matter of understanding that if I have a higher carbon dioxide, that means it was released. Because, photosynthesis does not release carbon dioxide, all right? But respiration does. If I see there is high carbon dioxide, that means there is more respiration. That means the pH is lower, we have more acid. Because more carbon dioxide is acidic. By the moment you find carbon dioxide is lower, that means it was used up by photosynthesis. All right? The pH will be higher. That means it is more alkaline. I think we can see it here as well. And get to memorize the colors. Yellow and purple. Mostly these are the important ones to know. Means if I have lower carbon dioxide, there is a lot of photosynthesis, the place is more alkaline. Yeah. Now, we have different tubes here, and also we have in leaves there. Here we have no leaf. The condition we have more light. If we have more light, we understand photosynthesis will happen and use up carbon dioxide. All right. If we use up carbon dioxide, the place is more alkaline. That's why it turns purple. For those who understood the previous slide of ours, I think we're easily understanding that. If we have a leaf in the light, that photosynthesis will happen, we use carbon dioxide. So we have less carbon dioxide. That means it will turn purple. There is a net intake of carbon dioxide. So photosynthesis is happening. If we put our plant in the dark, like I said, respiration will happen more. If respiration happens more, there is a release of a lot of carbon dioxide. If we release a lot of carbon dioxide, the place will become more acidic because the pH is lowered, then the indicators will turn yellow. There is a net intake of oxygen by a leaf. Now, the content where we have no leaf, we have light, then it will turn red. It is the other atmosphere we talked about. Then we understand that there is equal conservation. Now, this is how they asked this question. It was out of 6 marks, so we need to get to understand. Now we have our pH, color of indicator. Where we have pH 6.7, it's yellow. From talking about, you can already know what is happening here. It is yellow. Why? Why is the pH low? It is because you have more acid. All right? We have more acidic part here. Means we have more carbon dioxide. If you have more carbon dioxide, means more respiration happened here. All right? Because respiration with a lot of carbon dioxide. I think it is clear. Why do we have a lot of high pH? That means the place is more alkaline. We have less carbon dioxide. Because if you have more carbon dioxide, then we're expecting it to be yellow. So that means here we have less carbon dioxide. We can easily see that photosynthesis is happening here. Why? We don't have carbon dioxide. It's being used up by photosynthesis. Before you even read the question, you can understand that if you understand what you talked about. When is it red? When we have atmospheric level. That means photosynthesis and respiration, they're equal. Mostly on the daytime, not in nighttime, because nighttime we have more respiration. Now, I want us to understand this question together. Two test tubes were set up at the same time. Both algae and the hydrogen carbonate indicator were put in. At the start of the investigation, was red, because you saw that it is always red at the start in both. One test tube was placed in the dark. In the dark, more respiration will happen. Carbon dioxide will be released. pH will be lowered, like we said, because it is acidic, and it will turn yellow, like we saw. We have a lot of carbon dioxide. And one test tube was placed in the light. If it is placed in the light also, we should understand what it means to us. After 20 minutes, the content of the test tube in the dark were yellow. We understand we have less pH, high carbon dioxide because of respiration. On 6 marks, if you explain that on this, then it can be enough, surely. The content in the test tube in the light were purple. Why? Less carbon dioxide, all right? Because photosynthesis is taking place and using up the carbon dioxide. So the pH will be higher, and that's how we end up with the purple color, like we are from seeing here. Okay, guys, this is the answer here. For 6 marks, you can write at least this, okay? Photosynthesize in the light, where we had light photosynthesis had to happen, and carbon dioxide is used up. I think we understand that. It's why it turned purple. Means in purple, because carbon dioxide is used up, photosynthesis results in pH increase, and that's what pH indicator is telling us, and we have high pH. And in the dark, we saw that algae can only respire. Carbon dioxide is produced now, not used up. It's produced because we're in the dark, and in the light there is higher photosynthesis, and the higher photosynthesis turns yellow. Because remember, it is acidic, respiration is at the pH decrease, and it is more acidic, less alkaline, and become less neutral. I think it is easy to test this one of gas exchange, right? Understand, in the light, photosynthesis will happen, the pH will increase because we have less acid there, and that's how we're going to end up with with purple color. But in the dark, respiration is there, happening. With respiration, so you should understand what it means. Carbon dioxide concentration will be more because respiration releases more carbon dioxide and all that. All right, I hope it is clear. Something we need to know now, we are on extended term. We should understand the balanced equation for photosynthesis by using chemical equations. It's carbon dioxide plus water. And they like to ask these molecules a lot. How many molecules of carbon dioxide are needed to make one glucose molecule? Those ones are 6. And you see, this like equation is the opposite of respiration, because for respiration, now we start with carbon glucose plus oxygen, then these are the products. But now it is opposite on photosynthesis and plants making their own foods. So understand there are 6 molecules of carbon dioxide needed plus 6 molecules of water. That makes 1 molecule of glucose plus 6 molecules of oxygen. Light is converted into chemical bonds. Then we should understand in the bonds of holding the atoms, and we make 1 glucose molecule atom, which is here. We even have what you call the limiting factors. Limiting factor, because these are the needed materials, these 3: temperature, light intensity, and also carbon dioxide. One which is not enough might reduce the rate of photosynthesis, and that means it is a limiting factor. It is limiting the process. Now, if a plant is given unlimited sunlight, carbon dioxide, water, and it is in a warm temperature, the limit of the rate at which the photosynthesis is its own ability to absorb those materials and make them react. So we should understand that. So limiting factor can be defined. We should write this down in our book. As something present in the environment in such short supply, and it restricts life process. This plant might have enough temperature, have enough light intensity, but carbon dioxide, which is less, can be a limiting factor for photosynthesis. So they should be there on a good amount to make sure that the process happens. And that's when water is not in the limiting factor. We should also know that. It is not considered as a limiting factor, as the amount is relatively small. The plant does not need a lot of water. But these 3, any of them can be a limiting factor if it is in a short supply. and can restrict life, like we say. Now, starting with what temperature, let us see. Remember our previous video was on enzymes. Here what we explain, it is that the more the temperature, the rate of photosynthesis will increase. But it's until we reach here. We learned how to explain this very well. We saw that here, as the temperature increases, there is more collisions. Means these substrates are going to gain more heat energy, kinetic energy, that's going to help it move faster. Means they will increase the number of collisions between substrate and enzymes. At optimum temperature, we saw that we have a lot of enzyme-substrate complex, a lot of them here. But after the optimum temperature, the enzymes will start to denature. The enzymes will start to denature because the temperature is going to break them down. Like we saw in animals, like in humans, the optimum temperature is 37 degrees Celsius. Above that, enzymes start to denature. Same thing happens here. Means at optimum, there is more enzyme-substrate complex, but after that, we should understand the meaning. Like from what I told you in explaining this, we should explain from here. Come here. Say that the increase, the number of particles have more kinetic energy. There is more collisions between substrate and enzymes. This is optimum. We have more substrate and enzyme complex, a lot of them here. After temperature, you saw that there is maturation of enzymes. And reaching here, we have no activity. The word activity is given max 4, please. There is zero activity happening there. So that means we don't stop by saying the more the temperature, the higher the rate of photosynthesis. It means it's up to optimum. After optimum, there is the maturation of enzymes. I want us to follow nicely here. On light intensity, the more light a plant receives, the faster the rate of the process. But this one happens here. Means at low light intensity, there is an increase in the rate here. Okay? But until we reach here. Usually, we have light intensity, which is enough. The more we increase light intensity, the process is supposed to be, the rate is supposed to be increasing. But there is something important I want you to see here, guys. At this point, other factors becomes limiting. Means light intensity cannot be limiting because for it is increasing. But why is our rate of photosynthesis leveling off? Why does it level off? The problem is not light intensity because light intensity is keeping on increasing. The problem is other factors which are limiting here. That can be carbon dioxide, it can be temperature. Even in explaining this, the more the light intensity, the more the rate of photosynthesis increases until it levels off. But why is it leveling off? Light intensity is not the problem because it is increasing. The light intensity is influencing here, but here it is enough, but other factors are limiting. Temperature or amount of light, of temperature, sorry. Means factors which could be limiting when the line is on the graph is horizontal include temperature not being enough or not enough light. People, I hope this is clear because light intensity is affecting only here. Leveling off, it is not light intensity the problem. It is other factors that can be temperature or something else. Now, interpreting graph in limiting factors can be so confusing for so many students, but it is quite simple. In the section of graph where the rate is increasing, the line is going up, the limiting factor is whatever labeled on X, means here, from here to here, the limiting factor can be whatever is on X, like light intensity. But where it is leveling off, it is no longer this one. Now, in the section of the graph where there is not increasing, on the horizontal line where it levels off, the limiting factor will be something other than what is on X. So choose from temperature, light intensity, or carbon dioxide. By that time, for us, because you're talking about light intensity there, then the problem can be temperature and carbon dioxide. The moment maybe here we had like carbon dioxide and we see the leveling off, carbon dioxide is no longer the problem. The problem can be temperature and light intensity. This is how they ask them in IGCSE exams. State which factor could be limiting photosynthesis at X. At X here, what is limiting is light intensity. Because the more light intensity, the rate of photosynthesis is increasing. Starting from where we are leveling off here, the problem can't be light intensity because light intensity is keeping on increasing. Means for X, we can say it is light intensity. Okay? But for Y, What can be the problem? We look for other limiting factors because we saw 3 limiting factors: light intensity, temperature, and carbon dioxide. Means at Y, the limiting factors can be temperature and carbon dioxide. I hope it is clear. That's how we'll be explaining them. Now, coming to the leaf structure, which is the last part of our class today, I want us to know very well, guys. Take enough time to pause and get to understand all the parts of the internal structure of the leaf. Starting with the upper epidermis, and we have lower epidermis, we have palisade, spongy, and guard cell. I want to tell you that this light upper epidermis has no organelles inside. When I say it has no organelles, you can't find there anything like chloroplasts or other things. Why? This upper epidermis has an adaptation. It has to be transparent, so all the light that comes in enters. We have 3 parts: palisade, spongy, and guard cell. For those who remember what I talked about, you can remember what they have in common. Photosynthesis is exactly happens in 3 parts here. Photosynthesis, most of it happens in palisade mesophyll, but another photosynthesis also happens in spongy mesophyll. Photosynthesis also happens in guard cells. Why? We have chloroplasts inside here. We can see that. Palisade, they are rectangular. Means if they ask you the adaptation to photosynthesis, they have a lot of chloroplasts for photosynthesis to happen. If they ask us the adaptation of upper epidermis, which plays a very important part, they are transparent to allow all the light enter. Upper epidermis, lower epidermis, transparent. Palisade, spongy, guard cell, they have chloroplast. Photosynthesis can happen there. These ones are rectangular, these ones are round. And between the spongy mesophyll, we have airspace. And also we have what you call vascular bundle. The leaf, at the end of the day, we need to have water. Water comes via xylem. And also glucose, which is formed, we turn into sucrose and move from the leaf to other parts of the leaf. We should understand vascular bundles, the xylems and phloem as well. Means in the internal part of the leaf, upper, lower epidermis, 3 parts where photosynthesis happens, palisade, mesophyll, spongy, and guard cell. And at the end, we have the vascular bundles, xylem and phloem, where photosynthesis, where water is transported to the leaf via xylem from the roots, and also where glucose, which is produced, then later is turned into sucrose, which is going to be transported via the phloem. Even amino acid, we saw that it moves via phloem, by the way. This is what we typically think when we think of a plant cell, but it's just one of the type of cell around the leaf. And also you can understand that it is made from palisade. If you see, this is the palisade cell right here. So which adaptation does it have? It has a lot of, I think you can see a lot of chloroplasts for photosynthesis to happen in them. Guys, I hope you understand this because it is one of the most important parts, right? Upper epidermis, lower epidermis, where photosynthesis happens, palisade mesophyll, spongy mesophyll, guard cells, airspace, and also we have the stomata, by the way. Here it's where carbon dioxide enters through, all right? And we have xylems here, which are the big ones here, which brings water. Now carbon dioxide enters and water enters in the leaf. Now water will come in the chloroplasts and carbon dioxide, and also remember the chloroplasts have chlorophyll that can absorb sunlight. Now photosynthesis will happen. If you understand all those things, I think you are good to go with photosynthesis by now. We can see carbon dioxide entering. High concentration, and it is by diffusion, by the way, from high concentration to low concentration. Means high concentration of carbon dioxide in the atmosphere compared to inside means carbon dioxide is going to move inside. Carbon dioxide diffuses into through the open stomata, and we saw stomata, it is between the guard cells. The guard cells, remember, photosynthesis happens. I told you 3 places where photosynthesis happens: guard cell, spongy, palisade. And also carbon dioxide diffuses through the cell wall plus cell membrane. You can see it. And water already, remember, it is here in the xylem coming in. And light is absorbed by chlorophyll, which is in the chloroplast. Now all the needed materials are there, and photosynthesis can really happen again. So you will pause these and read them nicely, get to understand. We have a waxy cuticle. Waxy cuticle is here. Why is it wax? It is wax to not allow water to leave. Protective layer that prevents water from evaporating. We have the upper epidermis that I told you the adaptation. It is thin and transparent to allow light enter in the palisade. Mesophyll underneath it. And also we talked about palisade. We saw that they have a lot of chloroplasts to absorb more light and do photosynthesis. We saw spongy. Also they are round, but they have They even have airspace between them to allow carbon dioxide to diffuse, but of course they even have chloroplasts and all that. Lower epidermis contains guard cells and stomata. Guard cells, we saw that also, they open for stomata. They absorb and lose water by transpiration, but also open and close stomata to allow carbon dioxide diffusing. Should also understand that very well. And also we have the stomata where gas exchange takes place and opens during photosynthesis. Means during night, evaporation of water also takes place. It's via the stomata because it's an opening. Means water can go through. Vascular bundles, I told you these are xylems and phloem. Contain xylem and phloem to transport substances. Xylem that transport water from the roots up to the leaf, and also phloem that can transport glucose formed in form of sucrose to other parts of the plant, but also in phloem, so amino acid can go through there. We have xylem transporting water, and we have phloem transporting sucrose and amino acid. These ones we need to memorize them. I think it's near the same thing. Chlorophyll absorbing light. We have a network of venation that allow transport water in the leaf and carbohydrates. And also we have stomata that allow carbon dioxide to diffuse into the leaf and oxygen to diffuse out and all that. So I hope you get time to pause and get to understand. This question, I try to write the two here so we can see it very well. They can ask it in this form, and this form is the easiest because palisade, they are rectangular. Spongy, they are round and they have airspace between them. And also we have guard cells, we have vascular bundles here. We have chloroplasts inside here, which is the adaptations of them for photosynthesis to happen. But there are times they give you this one. Guys, you should be able to see it. If they give you this electron micrograph of a leaf inside, we should also be able to know it because they can ask it to you. So you should see these ones are upper epidermis that are transparent, like I said, thin and transparent to allow light to enter through. Light will refract inside and come in the palisade mesophyll for photosynthesis to happen. You should be able to see these round ones which are having space between them as mesophyll cells, spongy mesophyll. We also have guard cells, and the opening here, we have stomata there. We have waxy cuticle. It is wax. We saw that it prevents water loss. So you should be able to see the electron micrograph in this drawing. So like that, you'll be ready to know it in any scenario that they can bring it. Here is the question. That was asked in the past paper. Identify and explain how the structure of J and K are adapted to photosynthesis. J, we saw it. They are thin and transparent. I told you when I asked us about the adaptation, we should talk about the extension or the role and the role that part play. Very important. So if we talk about this J, we see that it is thin and transparent to allow light to go through inside. Okay. If we talk about K, these ones are xylem. Well, how are they adapted? The extension means they are thin and transparent to allow light to enter on the palisade cell. That's J, which is our upper epidermis. These are palisade, spongy. Now here we have our xylem. How are they adapted? The first thing, they are open end to end. It's like it is hollow to allow water to move from xylem all the way to the leaves. Another thing also, they are strong to give support to the plant. So those are the adaptations we can talk about. So we have another question here. These ones now, they should be very easy right now. Like I always tell you, when you see these kind of questions, read them with positivity because they are easy to do. If someone tells you have normal carbon dioxide and we have high carbon dioxide, we saw that carbon dioxide is needed for photosynthesis to happen. And as a matter of fact, what I want us to focus on, you will pause and read the whole question. I want us to focus on here. If they say concentration of starch, we see here we have 38 milligram per milligram of leaf, and here we have 67. What does that tell us? That tells us that where we have high concentration of carbon dioxide, we have more starch. More starch tells us that there is more high rate of photosynthesis because we produced a lot of glucose that was turned into starch. That means carbon dioxide, if it's less, it can be a limiting factor. Why is it limiting? You see, we produced less starch. We go to our question here. It was asked in IGCSE. I want us to read together. You will pause, maybe you first do it, then I put the answer there when you've tried. Now, shows the increase of carbon dioxide caused more starch. More starch tells that the rate of photosynthesis is high, produced in the leaf. This shows that at a normal carbon dioxide concentration, carbon dioxide is a limiting factor for photosynthesis. A normal carbon dioxide concentration, carbon dioxide is a limiting factor because you saw that less rate of photosynthesis happen, yeah? During photosynthesis, molecules of carbon dioxide are required to make one molecule of glucose. For those who remember what I talked about, how many molecules do we need of carbon dioxide? There are 6. Because so we need 6 molecules of carbon dioxide, 6 molecules of water to make one molecule of glucose. I hope you guys remember that, yeah? The greater quantity of starch stored in the leaf grown in high concentration, carbon dioxide concentration, means we needed more sucrose can be produced to transport in the phloem. That one we talked about it. We saw that the more we produce glucose, it will be turned into glucose to be transported. So the leaf acts as a what? If the leaf is the one producing glucose, and glucose will come from the leaf going in all parts, which name did I tell you that it is given to it? Means where glucose is produced, where things are transported from to where they are going, this is the source. Means it is the source. And for example, if sucrose is taken to the roots are called the sink. We studied that in the transportation of materials. Means more sucrose can be produced for transport in phloem. So the leaf acts as a source, and while we are sending it, we act as a sink. The greater number of chloroplasts per cell in a leaf grown in high carbon dioxide concentration means the more energy can be absorbed from and transported to which energy. This one also we talked about it. We saw in chloroplast the role of chlorophyll is to convert light energy into chemical energy. Means it's light and is transferred into chemical energy. The transpiration rate is lower when carbon dioxide concentration is higher. This means reduced loss of water from the leaf. That's what actually transpiration is there for. But if I talk about magnesium ion, guys, what comes in your mind? We saw 2 minerals, magnesium ions. We saw that they are needed to make chlorophyll to prevent the leaf from having chlorosis, which is turning it into yellow. And we saw that here the plant will turn into yellow because you have no chlorophyll. And if you don't have chlorophyll, there will be less of food produced. Then this plant won't grow. Means magnesium ion concentration is lower in those leaves because they have used the magnesium ions to make chlorophyll. I think that's clear because that's the only thing that magnesium is there for. So I hope these are the learning objectives. I want to think that if you follow all the video, you come and read and you see that if everything that you learned, you understood it. So starting from understanding the carbohydrates that are given out as a definition, we saw that's the process by which a plant manufactures carbohydrates, glucose, from raw materials using energy light. It's always having 3 marks. You should know how to write the word equation in words and the chemical equation of it. And remember how many molecules of carbon dioxide, how many molecules of water we need to make one molecule of glucose. There are 6 molecules we need to make one. We saw that chlorophyll is a green pigment which is needed to absorb light, and we saw that converts light energy into chemical energy. All those things that we talked about. This is other objective that we had to cover. Stating that most leaves have large surface area and are thin, and we should explain the features of adaptation for photosynthesis. We talked about the internal parts and everything. We talked about the internal part. We saw chloroplast. Inside we have chlorophyll. Waxy cuticle, we saw that it prevents water loss. Guard cells, photosynthesis happens there because it has chloroplasts and it's open for stomata to be there. And we saw stomata allow gas exchange, oxygen to come in and carbon dioxide to come in and oxygen to get out. And also we have spongy mesophyll, airspace, vascular bundles. We saw xylem and phloem. The adaptations of this one, we saw that it is hollow, open end to end to transport water. But for phloem also, we saw what phloem can transport, sucrose and amino acids. And we saw how they are adapted to their functions. If I ask the adaptation, like upper epidermis, we saw it's thin and transparent to allow all the light to enter. If they ask us the adaptation of palisade, they have a lot of chloroplasts to do photosynthesis, same as spongy and all that. So we saw what glucose which is produced is used for. We saw 5 points or 6. We saw it is converted into starch, it is converted into cellulose, it is used for respiration in plants, it is converted into sucrose, which is the way of transportation, and also it is converted into nectars, which is used to attract pollinators. We saw 2 ions, nitrate, which is needed to make amino acids. Remember, if I have glucose and I add on nitrate ions, then I'm going to make amino acid. I explained. We saw the N part, nitrogen, is the one we need mostly. Then we make amino acid, which is needed for growth. Magnesium, which is needed to make chlorophyll. We saw the investigation, the need for chlorophyll. We saw the reason why we start always by boiling, putting it in alcohol. We saw we boil to break down cell wall and cell membrane. We saw putting in alcohol. Then later we are going to see if chlorophyll is really needed for the light also. We saw it, that we take starch, and also for carbon dioxide. We saw when one is given carbon dioxide and another leaf is not given chlorophyll. We saw that the one that, carbon dioxide, we saw that the one with carbon dioxide, we find start there at the end, which is an indicator that photosynthesis happened. So you will get to come back. We talked about the limiting factors. We saw 3 limiting factors. That was temperature, light intensity, and carbon dioxide. We saw water is not a limiting factor. And in explaining, we saw when we have something that levels off, if I have carbon dioxide, means carbon dioxide control here, means the more carbon dioxide, the rate of photosynthesis will increase. By the moment we are here, because carbon dioxide is still increasing, it can't actually be a limiting factor. Means if it is carbon dioxide which is on X, then limiting factors here are going to be temperature and light intensity. Okay guys, that's it. I hope you will take enough time to watch fully the video, and you can even go to your learner's book and do different exercises to understand it better. But if you take enough of your time to watch this video very well, it's enough for you to understand everything that we talked about in photosynthesis, and like that it can be easy for you to answer each and every question that you be asked, whether it is in international exams of your IGCSE or even in your classes. So thank you guys. See you in the next one of tomorrow on the session. Bye bye.